Okay, and just a heads up then, so the meeting's going to be recorded. If you don't want to be recorded, just mute your video and audio. And it started.
All right, so this is the Coast to Coast Journal Club for, what are we today? The 8th of January, 2014, and we'll be talking about riboswitches today. So I send out a number of interesting references for people to look at. The main paper we'll be talking about today is genetic control by metabolite binding RNA, mRNA. And that is actually the paper that introduced riboswitches as a term in 2002. There were actually two other papers that same year that came out with discovery of riboswitches in other systems as well. So that was definitely sort of a development waiting to happen, essentially, and three different teams came out with results the same year. The other paper that I'm skimming over a little bit today is called A Decade of Riboswitches. It's one of the better review papers in terms of showing you the variety of things that RNA can do, and especially riboswitch systems. And I also, in the email that I sent out, I also listed three other papers. One is called Emerging Applications of Riboswitches in Chemical Biology. It's a somewhat older review paper. I think it's from 2010. Again, a fairly good review, has some much more understandable or much more easily readable figures in there as well, including talking about Salix, how to make aptamers, things like that. And then the other two that I have in the email that I sent out are two ways of coming up with sort of large-scale engineering approaches for riboswitches. So there's one called Versatile RNA Sensing Transcriptional Regulators for Engineering Genetic Networks. And that one actually talks about using pieces of RNA as the regulators. So they're using antisense regulation. So you can totally leave out the protein side. You can express an RNA and have that regulate other expression of other RNAs. And then the other paper called Higher Order Cellular Information Processing with Synthetic RNA Devices talks about a very interesting engineering approach in building logic circuits, essentially, in building Boolean gates, multiple input devices out of RNA, using a hammerhead ribozyme to which they attach various aptamers, and then you can get AND gates and OR gates and things like that. So it's a very nice engineering approach to developing new tools for synthetic biology using riboswitches. So, so you'll have an email with all those references in there. So let me start out with the main paper for today, Genetic Control by Metabolite Binding mRNA. So this is a paper in Chemistry and Biology from 2002. And I thought their introduction sort of made a good effort at sort of outlining the state of the field at a point where they sort of made their discoveries. They start out by pointing out that it was already known that RNAs can very selectively bind small molecules. And there's actually some technologies like Celix to develop new aptamers, so new RNA structures that are specific, that have affinity for specific small molecules. It was also already known that you can engineer ribozymes, so enzymes that are made out of RNA essentially, to be modulated by the binding of specific effector molecules, right? So once you have a piece of RNA that can bind a small molecule, you can use that to modify the behavior of an enzyme that's composed out of RNA, right? which led to a lot of people sort of asking the question, okay, what else can RNA do? Another thing that they're not mentioning here in this introduction, this paper, is the discovery of RNA thermometers. So that's a term that was coined back in 1999, actually. So you can actually have a piece of RNA that forms a secondary structure, sort of a stem loop structure, where that stem loop structure is actually sensitive to the environmental temperature. So if you raise the temperature, that stem loop structure will unfold and then might release a ribosome binding site and induce translation of a downstream protein. And this is actually one of the mechanisms that organisms sense temperature. Right? So it's a very short leap of logic from there to ask, okay, are there cases where we have one of those RNA structures maybe in an untranslated region of a gene, so in the 5' untranslated region or the 3' untranslated region, that affects the protein expression of that gene, and does so by directly binding to a regulatory small molecule. There were a number of people at that time looking into sort of metabolic regulation of various core metabolites, especially essential vitamins and things like that, looking for the protein intermediary that they thought might be involved in regulating some of these metabolic pathways, right? So often when you need to regulate the level of a certain vitamin inside the cell, you might have an option of synthesizing that vitamin yourself or trying to import it through a transporter from the outside world, right? And you want to balance those two pathways off against each other. So you want to sense how much of your vitamin is present inside. If not enough of the vitamin is there, you might want to import it from the outside. Or if you have enough precursor internally, you can turn on a biosynthesis pathway. Right? So a lot of people were thinking, okay, there must be some kind of protein involved that senses the level of that vitamin precursor, and then that protein then will act as a transcription factor. And in several cases, people were not able to find that protein intermediary. So people started looking at these alternative systems where these small molecules might actually interact directly with the RNA and regulating those pathways post-transcriptionally rather than interacting with transcription factor and regulating the expression of the pathway that way. So that's sort of that background in terms of RNA research. That is why essentially 3 independent teams all came up with the discovery of riboswitches in the same year. I could have picked any of the other 2 papers as well. I picked this one because it was the one that actually introduced the term riboswitch. So a riboswitch here essentially is it's a piece of the messenger RNA, typically, that will interact directly with a small molecule and turn off or turn on the expression of the protein, or the expression of the gene in general. The specific gene that they're looking at here is this BTUB gene in E. coli and Salmonella typhimurium, which is involved in regulating of some transporters for uptake of coenzyme B12 related compounds. And another paper had already shown that the presence of adenosylcobalamin actually inhibits ribosome binding to the mRNA for this particular gene, right? So they already knew that this gene was regulated directly by this sort of vitamin B12 precursor, and it was regulated at the level of ribosome binding, so post-transcriptionally in this particular mRNA. So these guys then started looking at direct interactions of that compound, adenosine cobalamin. If you look into introduction, the full name is there. It's 5'-deoxy 5'-adenosyl cobalamin, or ADO-CBL, which is the term that they're using to refer to it for the rest of the paper. I think I'm just going to call it B12. It's a B12 related compound. I'm just going to call it B12 for now, just to make the pronunciation a little easier. And I'll further on in the paper, they're discussing various other compounds that are related to that, and I'll try to distinguish which ones are which at that point. But for now, I'm just going to call it B12. So they say in the introduction, we speculated that the BTUB mRNA leader, so the front part of the mRNA, the part that's before the translation start site, so between the transcription start site and the translation start site, right? So that's the 5' UTR, the 5' untranslated region. So they speculated that mRNA leader might indeed be using an allosteric mechanism to control the level of translation in response to the concentration of ADO-CBL, B12. So an allosteric mechanism, in other words, that it would be sort of direct binding of that small molecule to the mRNA. And this decreased expression of the BTUB gene, which encodes a transporter protein for cobalamin compounds, is observed in cell-based assays when sufficient B12 is present. Right? So if you have enough of that vitamin precursor, you don't have to turn on the transport machinery. So that's the idea for this paper here. And let me quickly go over the section headings. It's actually kind of nicely organized in the sense that you can read the section headings and sort of figure out what's going on. So they start out with the first section on the results and discussion with metabolite-induced structure modulation of a messenger RNA. So they're essentially showing that the presence of this small molecule changes the secondary and tertiary structure of this messenger RNA. And then the second subheading there, 2 pages later, says a saturable metabolite binding site is formed by a messenger RNA. So this actually shows actual binding of that metabolite to the messenger RNA, and show that that is a saturable binding in the sense that if you add competing compound, essentially you can elute it out and things like that. And then the next section says the BTUB mRNA leader selectively binds ADO-CBL, so the vitamin B12 compound. So that's an issue of selectivity. It's not just binding all possible B12 compounds, but this very specific compound. And then the next step is then showing, the next page, disruption of metabolite RNA binding has consequences for genetic control, right? So they showed the presence of that small molecule changes the shape of the RNA. That small molecule actually binds directly to the mRNA. That binding is specific, and that binding then results in changes in the expression of, or the translation of the actual protein, the downstream protein. So that sort of wraps up the case for these riboswitches. There's a couple of interesting things that are actually not in this paper. They really don't talk about the mechanism of the regulation in here. They're just showing that you have some kind of change in the structure of the mRNA, and that results in a change of expression of the protein. But they're not even talking about how that riboswitch is occluding ribozyme binding site and things like that. So there's some interesting things that are actually missing in here that they probably could have added with a little bit more effort. But this is definitely a very hefty publishable unit already. So So let's dig into some of the specifics. Anybody have questions at this point? Feel free to interrupt me and ask questions. I talk way too much anyway. So
Yeah, Patrick, I have a question about the initial assay that they used, which is where they looked at the conformational changes. Now, so the types of assays that they use here are totally unfamiliar to me.
yeah.
So
They are pretty cool. Let me just dig into that section, and I'll go over those assays and what they do.
Yeah. Just briefly, it seems like this conformational change is it's basically the RNA itself is going to be spontaneously autocatalytically cleaving itself, and depending on how it's shaped, you're going to have different attacking groups positioned in different locations. And this looks like some kind of a surrogate assay for conformational change. Now, is this typical for folded RNAs to use this type of autocatalytic or assay? It seems to look at conformation change.
It is a technique I've seen used in other cases when looking at the RNA structure. There's some other assays you can do as well, like variants of the other ones that they're showing there, where you have maybe an RNase that cleaves specifically depending on the structure, so then you can use that. But yeah, you need to have some way of saying these things are base paired and these things are not base paired, and the easiest way to do that is by having an enzyme that cleaves base paired or non-base paired RNA.
Yeah, and I mean, I haven't done any of these assays before. Is there anything I guess more kinetic that you can do, non-destructive? Like, I don't know, I'm throwing this out there, circular dichroism, or which they do on proteins, or NMR or something like that.
I bet there is. I mean, if you know that the conformational change, for example, separates the ends of the mRNA, you can try and attach some chromophores to there and maybe see sort of do a Fred type assay, look at quenching, depending on the conformational shape and things like that. I haven't looked enough into the actual experimental assays for doing these things to know whether that is something that's commonly done. Again, this paper is from 11 years ago, right? There's a lot of groups that are working on riboswitches since then. I bet there are more sophisticated assays available to do those kind of things these days. But let me dig into that section and go over the assays that are used in here. So let's have a look at figure 1 in the paper. Let me see if I can pull this up on the screen here. Let's see if I can put this on my screen and then share it with everybody. That should be possible, right? Share screen. All right. Do you guys see the paper?
Got it.
It kind of screwed up my my screen here. I don't actually see what I'm sharing with you at the moment. There we go. Yeah, that screwed your image up as well. It's not supposed to look like that.
A little cut off.
You can only see a part of the picture. It's also screwed on our end.
Yeah.
Oh, now it's better.
Oh, it's good now.
Yes. Okay. All right. So over on the left there, they're showing the main assay, or the main assays that you're using to figure out the structure of this mRNA. And on the right is showing the inferred structure of the mRNA. And you see that this leader sequence, and remember, this is the 5' untranslated region. It's actually quite hefty. It's quite large, and it forms a fairly complex structure with lots of stem loops and a pseudoknot and things like that. This is not that unusual for natural riboswitches to be fairly complex. There are some much smaller riboswitches that people have engineered using CLEX, actually deriving aptamers that bind specific small molecules that are much more compact. I think the smallest one is something like 27 bases. So these things can be really tiny compared to the size that you would need for a regulator if it were made out of protein, right? Figure 1b, that structure, that is the structure as they thought it was at that time. Turns out that there are some mistakes in that error, in that structure, but they're not that relevant for this paper. You can look at some of the follow-up papers on this one to see the structure as people currently think it looks like. So just to orient you, there's I think there's 9 stems that are labeled P1 to P9, and all the way over at P9 on the top right there you see coding region, right? So that is where the gene itself actually starts, or where the codons for the gene, the coding region for the gene actually starts.
Patrick, how do they know? Very basic question. How do they know what which part is what on the gel? Like what? How do they know that fragment 2 is that part?
So let me get to what you see there on that gel on the left.
Yeah.
And I'll start with the column that's sort of the second from the right, labeled OH. And that is actually degradation of the mRNA using, what are they using? Potassium hydroxide, I think, which is something that's going to degrade the entire mRNA essentially. And before they do this degradation, they're actually attaching radioactive phosphorus to one end of the mRNA. I think it's the 5' end. That's is that correct? Yeah. So the 5' end of the mRNA they labeled with radioactive phosphorus, and then potassium hydroxide is going to bind sort of randomly throughout the mRNA. And what you're seeing on the band is just the fragment of the mRNA that has the radioactive phosphorus included in it, right? So the shorter pieces, the pieces that have been cleaved closer to the most 5' end of that mRNA are going to run through the gel farthest and form a band at the bottom of the gel. And then the longer pieces, including the intact mRNA, are going to be at the top of the gel, right? So that's why you see that beautiful ladder where each band in there really is cleavage at one of the individual individual bases in the mRNA. So you can literally, if you know where to start, you can literally count the number of bands there, and that gives you the individual bases in the mRNA, starting from the bottom, the shortest fragments. And then the column to the right of that, labeled T1, that is a digest with RNase T1, which will cleave only at Gs, right? So it's the same idea. You get the fragments that include that radioactive phosphor at the 5' most end of the mRNA, but these fragments all terminate in a G, right? And that's why you get far fewer bands there than in the one with potassium hydroxide, and each of those bands corresponds to a G in the mRNA. Right, so the bottom one there that's labeled G23, you can look on the structure where G23 happens, and it's actually the one that's got like a little light blue triangle with a 1 next to it. Can you see that in the structure? I can probably point at it. Can you see the cursor that I'm moving around here? Right. So that G here in red with the number 1 next to it, that's G23. And you actually see that there's, at position 22 and 21, there's Gs as well, right? So if you look back on the gel, there's actually 2 bands underneath that G23, and that's G21 and G22. So you can perfectly line up all the bands in that T1 column with all the Gs in the entire mRNA. And it gives you like a beautiful reference to where you are.
What I'm not getting is how do they know that for the plus and minus bands that those bands correspond to those particular Gs? How do they know that it's not some small? I guess I'm not very familiar with RNA mediated autocatalysis. Like, why can't it be some other G that's doesn't have the arrow pointed on the diagram? Like, how do I know it's that one particular G that's 23 nucleotides long and not some other bit somewhere else, I guess?
Yeah,
Like,
so these bands will migrate to their very specific positions, and they have single nucleotide resolution here,
right.
which you can tell from that column that's labeled OH.
Yeah.
You can distinguish between pieces of RNA that differ by a single base, right? And they know exactly how far those fragments will travel. And the bands are always the fragment of the mRNA starting from the 5' end of the mRNA to the cleaving side, right? Because the 5' end has that.
Why it's from 5' to the cleaver side? Why it cannot cleaver the 5' part of it too? You know, it can cleaver it from the middle, right? And then how can you deduce from the length that's what you're looking at?
Yeah. So the mRNA might be cleaved in 5 or 6 pieces, right? But only the part that has the 5' end has that radioactive label that will show up on the gel. So all the bands you see on that gel are radioactive label, right? It's not just presence of RNA. They're looking for the presence of the radioactivity there.
So when on the picture where you have 5' and PPP, it's where is radioactive phosphorus is attached, right?
Yes, exactly. Yeah.
And all other places don't have radioactive phosphorus, so if it's cut it off this end, you will not see it on the plus and minus bands.
Exactly. Yeah. So there's no way to know how many fractions this mRNA has been cleaved into. The bands will only show the fraction that includes the 5' end.
I have another question about this picture. If you look at the loop number 2, it has guanine bound to guanine, adenine, bound ACE to ACE, G to Gs. How could it happen? Like, well, why it's a loop? Number 2.
Which one is that?
Number 2.
P2?
Oh, it's not P. Yeah, it's P4. Next to P4, there is a loop. I don't know how it's marked. It's not marked, but I guess it's probably not a loop. They just drew it as a loop. Between P4 and P5.
This figure is not the best figure that I've seen for mRNA structures. It's kind of hard to figure out what's just drawn next to each other versus what's actually an interaction. You have to look at the little dashes that show the base pairing.
Still, if you look at P6, it has G bound to U's and U's and U's. This one. How could that one form a loop? How could you infer that one form?
Yeah, so there are some like wobble bases and things like that, especially when you're dealing with fairly contorted base pairings. You can get odd hydrogen binding essentially between 2 bases that shouldn't be interacting, like G and U.
Actually, Patrick, if you kind of zoom in close on that, it looks like in most places they're showing where you have binding, and in that particular area there is not binding. It's just a loop. I mean, it just see everywhere else it shows the bonds.
Well, so I'm not sure why they're not showing a bond between the C and G here. But there's a dot between the G and U, and there's a dot between U and G here, U and G here, and there. So these are non-canonical base pairings, essentially, where you still get hydrogen bond interaction, but it's not like a perfect GC interaction, essentially, or AU interaction. But everywhere where you see like the little dashes or the dots, you do have some form of base pairing, either a nice perfect base pairing or these sort of non-canonical base pair interactions. I keep thinking it might actually be better to maybe sort of draw some boundaries between the parts of the RNA that are not interacting, just to clarify this figure. I've definitely seen much clearer figures of these kind of interactions. So yeah, my apologies for picking a paper with a very confusing figure in terms of the structure. So I explained figure 1a, what this OH and T1 ladders are, and then the main data are in this plus and minus columns, the first two columns on that gel. And in these columns, they're not using sodium hydroxide, they're not using RNAs, they're just letting the RNA degrade all by itself, essentially. Right, so RNA is not terribly stable. If you just leave it at room temperature, even without having any RNases present, it will start breaking down. And it'll start breaking down first at the parts that are not base paired. Right, and that's literally what they're doing here. They're just making sure that they're working with very pure RNA, so there's no RNases contaminating things. And then they just let it sit for almost 2 days at room temperature, and then they come back to it and see where it's been cleaved. And that's literally what you see there. Those are the bands there that are most susceptible to that cleavage, purely due to the fact that the mRNA is not terribly stable and will break down all by itself. And then the plus and minus are simply in the presence of the small molecule, so this B12 compound, or in the absence of that molecule. And so you're looking for the differences in the bands between those columns. And those are the ones that put like this light blue triangles next to. All of the bands that you see in those columns are cleavage products. Not all of the cleavage products differ between in the presence or absence of the small molecule. Right. So in the figure in 1b, all the ones that are the bases that are labeled in yellow says cleaves with or without A2CBL. So that's all of the bands in those first two columns essentially. And ideally, when trying to figure out what the actual structure of this mRNA is, you can use that as a clue that tells you which of these bases should be paired and which ones should be unpaired. And you can tell they haven't done a perfect job here. There's definitely some base paired bases in here that are actually labeled in yellow. And that gives you one clue that their structure that they've written down here might not be perfectly correct. And if you look at the more updated versions of this imputed structure, they match actually much better with these labeled bases that should be unbase paired. But really, for the purposes of this paper, they only have to show that there is actually a structural difference in the structure of the mRNA with or without that small molecule. That's the only thing that they really have to show here. They don't really have to show where those differences land on the imputed structure. It's really sufficient for the purpose of this paper to show that there are structural differences that show up in differences in cleavage of the mRNA. And they can actually also use some of these bands to then quantify how much DNA has changed, right? So further on in the paper, they then look at different amounts of this small molecule they add, and they see what amount of the small molecule they have to add to have half of the total maximum amount of cleavage for that band, right? So that's one way that they're using to actually quantify the binding of the small molecule to the mRNA later on in the paper. So as I said, this is really only showing that there is a difference in the structure. You don't necessarily have to have figure 1b. You don't have to have the structure that they figured out from various other lines of evidence. It's really enough to have figure 1a to show that yes, there is a change in the conformation of the mRNA induced by the presence of the small molecule. All right. Let's see. Oh, just to go back briefly to that structure. You see that they have a section there that's labeled B12 box, right? And that, based on previous evidence, seems to be the actual aptamer. That seems to be the region that seems to be interacting with that small molecule. They don't go into detail into that previous evidence for that, but that's actually where the actual aptamer is. Those are the bases that interact with the B12. And presumably they figured that out by actually making mutations in that B12 box and showing that it destroys the binding with the small molecule. So, all right, so they've shown that you do get a structural change. Then the next step is to show that you actually have binding, right? At this point, if you show that there is a structural change in the presence of the small molecule, there could still be a protein intermediary, right? There could still be this vitamin B12 compound binding to a protein, and then the protein regulating the change of the mRNA. So next thing we have to show is that the small molecule actually directly interacts with the mRNA. And that's the assay that you're showing next. And unfortunately, there we go, yeah. So a saturable metabolite binding site is formed by a messenger RNA. So this they are doing, oh, and actually let me first show figure 2 here, where they're doing what I mentioned earlier, where they're varying the concentration of this B12 compound and then tracking specific bands in that gel from figure 1 to see how much of those bands are showing up. Right? And figure 2b shows that if you're looking at different bands, either the bands that increase in the presence of the compound or the bands that decrease in the presence of the compound, the concentration at which they reach the half max is about the same for all of those bands. So if you had 2 binding sites for the small molecule to the same mRNA, you might see 2 different classes of bands that behave differently at different concentrations. In this case, this conformational change seems to be seems to affect the entire mRNA in the sense that it affects all of the bands simultaneously with the same relation to the concentration of the small molecule. So that's something useful to know as well. It's also not a very sharp transition, which indicates that you don't seem to have cooperative interactions, for example. So you might need to have 2 molecules of that ADO-CBL binding at the same time. And in that case, you would see a much sharper transition than the fairly gradual transition that you see here. This half max also allows you to sort of figure out the affinity. So in this case, it's around how many, let's see. I mentioned further on how many millimolar. So it's a KD of around 300 nanomolar, which is actually pretty decent for small molecule binding a piece of DNA.
Hey, can you explain what it means, KD of 300 nanomolar?
Yeah, so that's the concentration at which you have essentially half of the amount of conformational change, right? So at some point, if you keep adding more and more of that small molecule, that change is just going to go all 100% to one conformation. If you don't add enough of the small molecule, have 100% of the original conformation. This is a concentration at which you have exactly half.
And how do they measure it?
They measure it essentially by that figure 2b. I don't know if they have any more.
No, I mean that figure 2b shows it, but how they measure the concentration, conformation? How they get those numbers?
So what you see on figure 2b, each of those dots is essentially quantification of the bands that you see in figure 2a, right?
Oh, okay.
So figure 2a, all those initial columns on the left they're like the two columns on the left of figure 1a, except rather than just absence or presence of the small molecule, they're using different concentrations of the small molecule. So they can see at what concentration the conformation switches from one state to the other.
No, I understand, but how in practice you don't watch with your eyes, right? It's how by brightness of the band, or how you compare those things.
Yeah. So figure 2a is just two strips that they've cut out of a gel that's very similar looking to figure 1a. They just have more bands with different concentrations, essentially. Mm-hmm. But they do exactly the same kind of assay. You have the ladder with potassium hydroxide. You have the ladder with RNase T1. Those are the ladders on the right. And then you have all the bands with just mRNA spontaneously degrading in the presence of different concentrations of the small molecule.
Yeah, I got that, but how you get the number like 0.25, 0.50, 0.75 fraction cleared? How you get those numbers? You just compare
by quantitating those bands, right? So you have to. That's by quantitating how intense those bands are, right? So that's just some image processing on the gel. See how intense of a black color you get there, and you try to quantify that.
Okay. Thank you.
So then the next step.
All right, Patrick, I got a quick question. So we're looking at that figure where you have figure 2 and B, where you have all those the log C and the fraction normalized and 1, 2, 3, 4, 5, 6, 7, which are the different. Those are the different. Cleavage sites, right? The pieces, and they're kind of like overlapping everything. So there's no error bars in there. There's like circles and squares. I mean, how I haven't done any of these KD studies, to determine the dissociation constants. I mean, is it typical to kind of like look at all these sites and kind of like aggregate all of this data together, or is it more typical to look at each site individually and because I'm looking at all of these, I'm trying to like squint through all of this data and it looks like, if you overlap everything, you get this kind of linear correlation and you can get this kind of KD, like right where X marks the spot where you get half fraction cleaved, or uncleaved. But then if you look at some things like the diamonds, it looks like you almost get you almost look like you get these exponential peaks, like a S-shaped curve. And I don't know if you're like losing some data by doing that, or that's typical to just look at all these multiple sites and aggregate it to try to get a KD. I mean, what's your opinion?
Yeah, it's a good question, and I'm not sure if they go into that. I don't think they mention it in their method section of exactly how they come up with that number.
Yeah. I mean, I get how they get the KD based on all of this thrown together on this graph, but I'm wondering if you're not losing some information because, you have all of this and, it looks a little messy. I don't know what the bars are. I'm sure they're all over the place. And you could see just from some of this stuff looks pretty linear, like the circles and maybe the squares, but then other stuff like the diamonds certainly does not look very linear, and so it's kind of hard to tell what's going on.
Yeah, they completely glossed over how they did that in this paper. The way
you need to keep in mind that the concentration here is logarithmic. So if you really expand it to non-logarithmic, you will have no spikes. You see those spikes with diamonds because it's logarithmic. It's very compressed.
Okay.
If you decompress it, it will be really, shale line. There will be no spikes. So, and that's why I think they can just grab it in the middle, because there is no precision here. It's like every step is 10 times bigger than the previous step. It's just an order of magnitude they give you. That's all it is.
Yeah, and that's really all you need, right? I mean, do you really care that it's 300 nanomolars versus 250 nanomolars?
Yeah, I don't know if 300 nanomolars is correct. I would say it's, hundreds of nanomoles would be a more correct answer on that scale. Yeah. Just saying that it's 10 to the -5, that's all we care.
And if I were to do this analysis, I would probably overthink it and trying to fit like a sigmoidal curve to each of these individual bands, and then calculate the predicted KD for each of the bands, and then take an average across those, something like that. It's probably overkill for what you need there. You just want an order of magnitude to know how good an affinity that you have, really. All right. So let's move on to the next section.
I'm sorry, I have a question about this 300 nanomolar. How it compares with proteins that you control the expression of genes? I mean, is it higher concentration, lower concentration, order of magnitude? Does anyone know?
It's like right in the middle, I think, right?
Compared to small molecule binding to a protein, I guess. Anybody?
Yeah, there's a chart. There's a chart out there we can pull out, and I think it's right. I think it's right in the middle. It might not be very. I think it's fairly tight binding. I think the range for intermolecular interactions is from the micromolar to the nanomolar range. Picomolar is very, very, very tight, and that's kind of rare, I think. There's a few things like streptavidin and biotin which go into the picomolar KD, but I think nanomolar to micromolar is actually pretty. It's what you would expect for intermolecular protein interactions.
Okay, and this is 0.3 micromolar, so right in the range what you're talking about. Okay.
Yeah.
Yeah. All right, next section. Saturable metabolite binding site is formed by a messenger RNA. And here, again, it's a pretty interesting assay that to pull out here. They're actually using a dialysis apparatus here. So, meaning they essentially using an apparatus that has two chambers that are separated by a dialysis membrane. And that membrane will allow the small molecule to pass through, but won't allow the mRNA to pass through. Right? So they can, by using that, they can figure out how that small molecule is going to equilibrate between the chamber that has the mRNA and the chamber that does not have the mRNA. Right? So in each case, they start out with the RNA. RNA in chamber B, I think, in each case. And they add the small molecule labeled with tritium so they can measure it easily in chamber A. And then they let it equilibrate a little bit, and if the small molecule actually binds to the RNA, it's going to wind up all being in chamber B eventually. If there's no binding, then the small molecule is just going to equilibrate between those two chambers, and you get the same concentration in both. Right? So it's a cute little assay that relies directly on co-localization of the small molecule with this aptamer RNA. Essentially, it binds the small molecule.
So, but okay, can you explain a little bit? What about proteins? Will it let proteins through?
There's no proteins whatsoever here. right?
How they achieve that? How they make sure that they didn't get on any protein that can bind to the small molecule and RNA?
This is totally synthetic, I think. This is an in vitro
Yeah.
assay.
Yeah, this is an in vitro expressed, in vitro transcribed RNA.
So it's purified components.
Yeah.
Oh, so you could. I'm sorry, probably my ignorance about how you can do it. So you can purify DNA, get all the proteins and everything, and get the pure DNA, and then express the DNA into RNA, translate it in RNA, and use it for this kind of experiments. That's what they do.
Exactly.
Well,
Yeah. So you start off with the DNA and RNA polymerase, and you produce RNA, and then you get rid of the DNA and the protein, and you purify the RNA, right? And I didn't read exactly sort of their protocol for doing that, but I assume they're using proteases to make sure all the protein is degraded and everything.
Okay, and then
this involves protein anyway.
Okay, and then sorry, go.
I just said I assume they just synthesized everything.
Okay, and then this experiment shows that RNA binds to a small molecule that no proteins are involved. Is that correct?
Yes, so they try to get everything as pure as possible. And I guess they can quantify how much protein might still be contaminating their RNA and all of that. I assume they've done all those checks to see how pure the RNA is that they have.
Yeah, I assume they just, I haven't read, I don't know what it says in the materials. I just assume that everything was synthesized, you know, like the cyanocobalamin and the labeled and unlabeled, you can get, those are just chemicals, and the RNA, I just assume they just synthesized that chemically because it's only 202 nucleotides.
No, they in vitro transcribe it from DNA.
Okay.
Because, I mean, we've seen in figure 1 and 2 that RNA will degrade on its own, right?
Right.
So if you were to order it synthesized from a company, if it takes 2 days until you do your experiment, then your RNA will be degraded already.
So nobody synthesizes RNA, right?
I think there's some people doing it for special purposes, but I mean, typically you just start from the DNA.
Okay.
Could
you just freeze it after you synthesize it?
Yeah, you can definitely put it on ice or put it in -80. All right, so some of the other things that you find in that figure 3 there is then looking at what happens when you add unlabeled small molecule, unlabeled ADO-CBL. And if you add a large amount of the unlabeled ADO-CBL, it's going to outcompete the labeled ADO-CBL. And that's going to make the label essentially equilibrate again between the two chambers. Right? So that's what they show in, let's see, panel, is that panel 2, I think, right? In figure 3. So they show on the left in panel 2, you have the labeled small molecule in A, the RNA in B, and then after 10 minutes they add, or then they add the unlabeled small molecule in excess, also to chamber B, and they see that this ratio of the label drops from 2:1 back down to 1:1, right? So you've been able to compete the labeled small molecule using an excess of unlabeled small molecule.
So Patrick, just make sure I'm understanding this. So the kind of the blue squares along the top with the little arced arrows are describing the multiple steps in the experiment.
Yeah.
And then the little moving dot on the line is their observation of the ratio of the labeled ADO-CBL after equilibrium is reached. Is that correct?
Yes. So each of those blue rectangles is essentially one data point from their dialysis apparatus. So in panel 1, they're showing just the labeled small molecule added to chamber A, and then you see where it says elapsed time.
Yep.
Right. Elapsed time 10 minutes. After 10 minutes, the label has equilibrated such that you see the same concentration in chamber A and chamber B. Right. So even though you added it to chamber A, after 10 minutes, it's in both A and B at same concentration level. Right. Then in panel 2, the blue rectangle on the left there, that shows the basic experiment where you have the labeled small molecule in chamber A, the RNA in chamber B, and after 10 minutes, you get 2 times more of the labeled small molecule in chamber B than in chamber A, where it was originally added. Right.
And presumably then that's because some fraction of that, 30% of that, I guess, is bound to the RNA that's tied up and can't get out of the B side.
Yes,
Is that
exactly.
right? And the other two, 66% is distributed evenly between the two.
Yeah, yeah.
Okay.
And then the on the right side of panel 2, they then show that when you add the unlabeled small molecule also to chamber B, then the label concentrations again equilibrate between chamber A and chamber B, because you're adding much more of the unlabeled small molecule and it just outcompetes the labeled one.
So that displaces the labeled one from the RNA?
Yes.
Okay.
Yeah.
Well, what the point of this, step? Why would you do that?
Well, it shows that it's not covalently bound, for example, right?
Okay.
So it's sort of titrating the
Is everybody else still here?
et.al. That does not compete with the et.cbl, right? So that shows specificity in a sense, right? So at least with that one particular other B12 related compound. And then further on in the discussion, they're talking about wouldn't you expect to see more than a 2-to-1 ratio between B and A? And what's happening apparently is that some of the RNA is already being degraded. And by flushing out the buffer, let me see if I can remember exactly how this worked. They essentially are removing some of the degradation products from the degraded RNA, and they can actually increase the ratio of the signal, essentially by cleaning up their RNA and getting removing all the inactive RNA. That makes sense? I'm not sure I explained that very well.
Not really. When you remove degraded RNA, the ratio should go down because then you will have some small molecule freed up.
Well, let me leave back to where this is described in the text. It made sense when I read it, but I haven't.
Yeah, I think you had that a little backwards. I think it's degradation of the B12 derivative.
Yeah. So it's on the next page, bottom left of the page. Assuming that the 1-to-1 complex is formed between effector and RNA, we had expected that equilibrium dialysis would produce a CPM ratio of far greater than 2 under our assay conditions. So they're having twofold excess RNA over 8-OCBL and concentrations of RNA and effector in excess of the apparent KD. So knowing that KD means that they know how much of the small molecule to add to really saturate the amount that's present. In this case, they actually want to add, let's see, excess of the apparent KD. In this case, they would actually want to add less of the small molecule than the KD to make sure that all of the small molecules bound by the RNA, and there isn't, and the RNA isn't saturated essentially, right? So then they go on, since there should be an excess of binding sites, the majority of the tritium, so that's a label on the small molecule, should be shifted to chamber B upon equilibration. However, our data suggests that about 70% of the tritium in the sample used is not in the form of, it's actually the labeled small molecule that is losing some of its tritium, essentially. So some of the tritium winds up essentially exchanging with the water, with the hydrogen in the water. So you just have tritium floating around in the buffer, not attached anymore to the small molecule. And that's why you don't see like a perfect, all of the tritium in chamber B and nothing in chamber A. So I had the explanation exactly backwards. It's not the RNA floating, it's the tritium that's falling off of the small compound. So, all right.
Hey, Patrick, can you explain what the that last experiment 4 there with the 4 steps? What were they trying to prove there?
Yeah, so that is where they're replacing the buffer, and so they're getting rid of all this tritium floating around, and they're essentially flushing both chambers A and B with fresh buffer. So they're getting rid of all this tritium that was floating around, and the labeled and unlabeled small molecule is still bound to the RNA, right? And you see that when you do that, you don't see the concentration of the label in B rise. Presumably what you're seeing is the concentration of the label in A drops to almost 0. Right? So they're getting a ratio of the label that's almost 4-fold as opposed to only 2-fold by getting rid of this free-floating tritium in the buffer.
What it proves?
It proves that you actually get a much better co-segregation of the labeled small molecule and the RNA than you would expect from just seeing that ratio of 2-to-1. It's more like 4-to-1, which is much more in line with what you expect in terms of sort of concentrations based on the number of binding sites that are available for the small molecule.
Although they got a ways to go, since you would expect a ratio of like 1 to 10 to the 7, right?
Sorry?
They got a ways to go to clean the chambers out, because you would expect a ratio of like 1 to 10 to the 7.
Yeah, and presumably that tritium is constantly falling off of the small molecule anyway, right? So when they replace the buffer, they have to make sure to take the measurement fast enough, or the tritium is going to go back into chamber A from chamber B. Right?
In the dark, supposedly.
Yeah. So these are not necessarily easy experiments to do. I mean, for one, in both of these assays, you're working with radioactivity, radioactive phosphorus and tritium. So these are not terribly DIY-able assays necessarily. So it would be interesting to look at some of the more recent articles on how to work with riboswitches and what assays are being done that are more amenable to something we could do.
Yeah, but why it's hard? You're saying that radioactivity does mean by itself it's hard. If it's small amount of radioactivity, it's irrelevant.
Well, it's hard to get a hold of, and there's more regulations around it. I know that, for example, at work at Lawrence Livermore, everybody's complaining that they can't really work with radioactive phosphorus because there's so many rules around it that people just give up, not want to deal with the red tape.
Tritium as
So
well.
you cannot get like 50 milligram of radioactive phosphorus without any proof.
Well,
Not easily.
I don't know how it is now, but I mean, I worked as an undergraduate in the early 1990s. I mean, at Hunter College here in City University of New York, I mean, everybody was using, you know, P32 and tritium, and I mean, it was every lab had a hot bench. I don't know how it is now, but I mean,
Yeah, no, this is the kind of work that if like an undergrad is being careless, the entire lab might get shut down for radioactive contamination. This has happened in several labs where somebody did a check with a Geiger counter and found radioactive traces all over the place.
yeah, you need good protocol.
Why it's relevant? It's because it can contaminate other experiments. Because you know, by walking on the street, you got a lot of radioactivity beamed into you. It's not like, you know, we live in radioactivity-free environment. You know, why it's so.
You don't have to convince us, Dennis. You have to convince Environmental Health and Services at the university.
Okay, so it's purely political and public issue. It has nothing to do with real science, and why there is no really need to be so clean. It's just
Yeah, it's state and local health regulations. You have to abide by the protocols.
And also buying radioactive compounds has certain levels of red tape attached to it as well, right?
No, I understand. That's what I was curious, because you know what, you drink water, you can bring your Geiger counter, and second cup will trick the counter. You know, it's everywhere. That's what I was wondering about. Yeah. Okay.
All right. So they've shown that the small molecule actually will co-segregate with the mRNA, so that you have a direct binding to the mRNA essentially. Then in the next section, they're showing that is a specific binding for that particular compound. And they've already shown a little bit of that by that panel, let's see, figure 3, panel 3, where they added cyanocobalamin and show that does not compete with the et.al, right? So in the next figure 4, they're going to into a much larger range of different compounds that are related to vitamin B12. And as you can tell from figure 4A there, B12 is actually a fairly large molecule, fairly complex, and it has various side chains. And you can have different things on those side chains. And if you look at the metabolic biosynthesis pathways for B12, there's all kind of different steps, attaching something here and then attaching it again somewhere else. So there's lots of different intermediates in the synthesis of vitamin B12. And they're using 11 different compounds here. And for each of those, again, they're doing their original assay where they're looking at one of the bands indicating a conformational change of the RNA in the presence of those compounds. Right. So if you're looking at the gels in figure 4B, each of those, again, is like figure 1A, essentially, where the bands on the left are spontaneous cleavage after a certain amount of time in the presence of these different compounds. And then those two ladders on the right in figure 4B are again, degradation at all of the individual bases using potassium hydroxide, or degradation only at the G's using RNase T1. Right. So they're focusing on the single band here, and they're quantitating how heavy that band is, how much radioactivity you get from the radioactive B12, the radioactive phosphorus here. And so the first band that you're seeing here, the leftmost band, that is using et.al. So that's the compound we've been using in the rest of the paper so far. And then they're showing that with these other compounds, 2, 3, 4, you pretty much see no cleavage or no change in the conformation in the presence of those compounds. Compound 5, you see a little bit, 6 a little bit more, 7 a little bit, 8 you have some reasonable cleavage, 9 less, 10 a lot of cleavage, and then you see 1 again, which is again our 8-OCDL. So you can figure out what is actually correlated with this conformational change, depending on what side chains are present on these molecules. And I'm not going to go into the details of this. Suffice to say that you could do this in much more detail and add many more compounds to test, and then really try to nail down sort of what parts of this large molecule this aptamer seems to be looking for to induce the conformational change of the mRNA. But you really only have 2 compounds in this list of 11 where you get very good cleavage, and that's 1 and 10, and they're actually very closely related to each other. So in 1 you get 5' deoxyadenylyl, in 10 you get 2' 5' dideoxyadenylyl. So that's the only difference between the 2 compounds, and they seem to behave almost equivalent in terms of inducing this conformational change.
So I was reading about the vitamin B12, and it seems that most of the food additives, they contain no actual vitamin B12, they contain cyanocobalamin.
Yeah.
If human cell uses the same mechanism to control transport of B12 vitamin in the cell, it means that when you get vitamin B12 in the form of cyanocobalamin, I'm sorry if I pronounce it wrong, then this mechanism will pass it through, and you can get your cells oversaturated with B12 vitamin because this mechanism will not be able to block it.
Yeah, so one question I would have, which is sort of a higher level question, is why does this switch need to be so specific for that particular B12 related compound, right? Seems like, can't you just measure all of the B12 precursors, and if you have one of those, you can make B12?
Yeah, I mean, if you read the Wikipedia page about B12, it says that cyanocobalamin, and correct me if I'm wrong, if somebody knows how to pronounce it right, it doesn't occur in the nature. Probably that specific compound is the only compound that other organisms, express into the environment, or when you eat something, it's abundant form. My question is, like, if we get in B12 vitamins in the form of cyanocobalamin, we're probably getting a toxic amount of it because this mechanism doesn't control it. I'm just curious.
So these riboswitches are from E. coli, right?
Yeah, I'm curious if we have the same one. That's my question.
Yeah, so E. coli actually does have a pathway where it can produce its own vitamin B12 from scratch. But it's a very lengthy pathway, and it's presumably fairly expensive to do that. So if it can import it, it would probably prefer to import it, I would assume. So maybe what it's sensing here by being so specific to this very specific compound here, maybe it's just checking for one very expensive step and uses that as its decision point of whether it should try to import from the outside world or synthesize it itself. So anyway, that's sort of beyond the scope of this paper, really, sort of asking why it's so specific. But it's interesting to see that even though these are all very closely related compounds and very large compounds, only differing in some of the side chains, there is very significant specificity here in the interaction with the mRNA, with the riboswitch. So, all right. So let's skip to the last section in this paper, and I'm going to cover this in as quickly as they're doing, They go into very little detail on the last part, actually showing that you have genetic control due to this conformational change, due to binding to the small molecule. Essentially, what you're doing is they're attaching a reporter gene to this mRNA leader. So they're engineering a reporter gene that has this piece of mRNA, this riboswitch in its 5' untranslated region, and then it's just looking for the expression level of that reporter gene. And then they're showing that, so as the title for this section says, disruption of metabolite RNA binding has consequences for genetic control. So you can show that the changes in structural conformation of the mRNA are associated with expression levels of the downstream gene, and you can show that if you make specific mutations in the mRNA, you can inhibit the regulation of a downstream gene. Right? So that's fairly straightforward. And again, we could go into this in more detail, but they're sort of glossing over that part as well in the paper. And I think I'm going to do the same in favor of switching to the review paper of riboswitches and showing some of the other things that you can do with these things. Any more questions on this paper in particular? So we've gone sort of very deep into the technical details for this, but I thought it was kind of interesting to do that.
How do you know that riboswitches are actually controlling expression of some genes and they are not just a noise, unavoidable noise that the cell has to deal with? How do you know it's something useful, not a noise?
Well, I mean, in this case, they're using an actual reporter gene to show that you can switch off or on the expression of that gene by controlling the structure of that riboswitch, essentially.
No, I understand, but evolutionary, why we jump into the conclusion that it's actually useful control, it's not some kind of a noise that got triggered accidentally and cell just has to live with that? And how do we know that evolution could use it, to really use it?
Well, in most of the cases where these riboswitches have been found, they've been found in the context of switching on and off biosynthetic pathways for small molecule, and the riboswitches have affinity for a related small molecule, essentially, right?
Yeah,
So,
but that can be a coincidence, because if you have I don't remember, 12 compounds or so related to vitamin B12, and only a couple of them turn on this switch, could be an indication that it's just an accident. I mean, it just, evolutionary accident that got preserved, not because it's useful, because it's got preserved.
yeah. So these things are evolutionarily conserved.
Oh,
And
okay.
actually, in some of the papers looking further at this BTUB gene and the et.al riboswitch, they're looking at all of the genes from other organisms that are involved in vitamin B12 synthesis or transport, and have an extensive 5' UTR that might house a riboswitch. And they do find that some of these structures are conserved. So there's evolutionary conservation. There's some of these things are conserved across the different branches of the tree of life. So they seem to be fairly ancient. And there's all kind of sort of speculation as to what this means for sort of the RNA world hypothesis of the origins of life.
Yeah, but it still proves nothing that it's conserved, because it could be conserved because mutation to it itself will block the expression without any small molecule being involved. It's a small molecule, you know, modified the conformation and prevents the expression the same way any mutation to this chain can change the conformation and prevent the expression, and that can be reason for conservation. It just could be a noise that you cannot change.
So I'm not sure I follow your point. Are you asking whether there might be some other regulators involved? Or?
Can I make a comment here? I think what you're
Sure.
getting at is there's a lot of stuff, a lot of detritus that organisms pile up internally and then have to deal with. I guess the answer to your question is, I mean, if you make genes, you know, if you take an organism that has known riboswitches and you get those riboswitches out, according to your hypothesis, the organism should function better, because you've gotten rid of that noise, or it should not affect the phenotype negatively.
Yes,
right?
yes, but no, don't mutate. Just cut it off completely, because mutation can have the same effect as you know,
Okay.
Being regulated.
Right. So okay. So scramble it or remove it out or change that leader sequence. So your E. coli that has you know, your known riboswitches. So you generate an E. coli that doesn't have these riboswitches. That's what you're saying, right? So and then basically compare it to one and see whether or not in a certain environment it behaves at least equally, if not better, than the one with riboswitches.
Yeah, that would be a correct experiment. That I agree with you. I just wonder if somebody did or if somebody would.
But you can make that argument for any kind of regulator, right?
Yes, yes, because just saying that it's it has useful function because it's well preserved, it's not correct statement, because it can be well preserved because messing with it will break the stuff that got expressed. You know, that's my point. It's just by saying that it's well preserved doesn't mean that it's useful. You know.
Well, we do know that messing with this region sort of dysregulates this particular gene, which, I mean, they don't show it in this paper explicitly, but presumably has sort of a cost on the organism in terms of metabolism of biosynthesis of vitamin B12.
Yeah, and just to make one more comment that relates to what you said, Dennis, there's a review paper out. I don't know. I don't think it's the Decade of Riboswitches. Maybe I'm getting my wires mixed up here, but I don't know, Patrick. Maybe you read this somewhere before. Isn't there some experiment where there's an antibiotic that's hits a riboswitch that basically is it's not the B12 derivative. I think it's that thymine pyrophosphate riboswitch, and there's an antibiotic that mimics that and has a negative effect when it binds to that riboswitch.
Yeah.
So I'll find that review paper, but you know, I didn't read that primary paper, but there was a couple of sentences that they cited, and then the author went on to speculate that maybe some antibiotics have this effect that they actually target and inhibit these riboswitches.
Because these riboswitches are so pervasive in bacteria and in pathogens, they've actually been used as target for novel types of antibiotics.
Right. So that would.
I'm sorry for being annoyed, but not that. Again, it can be a noise in DNA that antibiotic accidentally triggers, but it doesn't mean it has a function.
But if you can disrupt a riboswitch using a piece of DNA, or antisense RNA for that matter, and it kills the pathogen, that seems to indicate that that's important for the pathogen at least. Right?
No, it just indicates it there. And it indicates you found a way to break it. It's just hair trigger that you found. It doesn't mean it was useful.
Well, it means that it's correlated with fitness of the organism.
No, I understand, but you know, just want to make sure we don't. You know, I want to make sure for myself that I don't treat correlation as causality, because two different things. And I think I found the paper. It says riboswitch control of aminoglycoside antibiotic resistance. So, yeah. Okay. Yeah.
So these riboswitches are by now well enough understood that we can really rely on their functioning to actually do engineering. So I think that's the important part. I don't want to get into the questions of why these riboswitches are there and sort of why these organisms are using them. The important part for us, I think, is these things can actually be engineered and can be used to make genetic circuits in synthetic biology that rely only on processing RNA and DNA, essentially. So this essentially allows you to make synthetic biology constructs that don't even have to involve expression of protein anymore. So let's quickly switch to the
Hey, Patrick.
the paper that I wanted to look at.
Patrick, can I ask one real quick question?
Sure.
So I think you might have referenced it early on in the paper, but I just want to make sure I understand. So the point is that when you have these RNA structures where you have segments of RNA that's not bound, those degrade more quickly. Is that correct?
Yes. So the parts of the RNA that are not base paired will degrade faster than the ones that are base paired.
Okay, great. That's what I assumed.
By changing that, you can see a conformational change essentially in the structure of the RNA.
Right. Okay, thank you.
So even without knowing what that structure really is, just sort of the appearance and disappearance of those bands tells you there is a conformational change. There's some things that were base paired in one conformation and are no longer base paired now and vice versa.
Right, thank you.
All right, let's switch to this other paper, Decade of Riboswitches. So this was 2002, and as I mentioned, since then we've learned a lot more about these riboswitches, and there's tons of different mechanisms. Till what time were we supposed to go for the journal club? Just a quick check. Were we supposed to end at 7, or doesn't matter?
We tend to vary quite a bit.
All right.
I think it was originally an hour, but we've gone longer in the past.
Yeah.
So-
All right. Let me quickly sort of run through sort of this figure 1, the diversity of riboswitch mechanisms. So this first paper that we did is sort of the canonical model of a riboswitch. You have something that's in the 5' untranslated region of the mRNA. So when the mRNA gets transcribed, it folds up into some structure that then can interact with a small molecule, and then typically sort of releases a ribosome binding site or hides a ribosome binding site and affects the translation of the downstream gene. So that is sort of the canonical model of how riboswitches work. There's many other types of riboswitches as well. So you can either inhibit or activate translation by hiding or revealing a ribosome binding site typically. You also have riboswitches that act at a transcriptional level. So this is actually co-transcriptionally. As the mRNA is being transcribed, it forms a 3-dimensional structure that can actually terminate transcription or halt transcription or pause transcription, whatever. And those riboswitches can also be regulated by small molecules. So now you have a case where you have a riboswitch that can act purely at the transcriptional level. You're not even talking about translation of a protein anymore. And then the downstream gene could be an RNA gene, right? So if the binding of a small molecule to the riboswitch sort of anti-terminates transcription, the gene being transcribed might actually be a functional RNA that then does something else, maybe even regulating other riboswitches. So figure 1A sort of shows all these different mechanisms. So you have translation inhibition and translation activation. You have transcription termination, which typically involves a stem loop followed by a bunch of U's, and that forms a transcriptional terminator. And if you can make a conformational change that then hides some of these U's or disrupts the stem loop, you can relieve that termination essentially and let transcription proceed. So you can have row-independent or row-dependent terminators, and you can get anti-termination as well. So that's transcription and translation. You also have figure 1b here. You can have ribozymes, I mentioned that earlier, that change conformation and then become active or inactive depending on whether they bind a small molecule. So these are things like hammerhead ribozyme, for example, will actually cleave its own RNA depending on whether or not that small molecule is present. So let me see, hang on a sec. Can you guys still see the figures on the paper?
Nope.
Seeing sharing is paused. Bring your shared window to the front. I'm not sure what that means. Let me try and turn off sharing and turn it back on. There we go. I think you can see it now, right? All right. So this is a mechanism you can use in various ways. So if you have this ribozyme structure that cleaves its own RNA, that cleavage again could reveal a ribozyme binding site. So again, you might have post-transcriptional regulation here that affects the expression of the protein. You can also have these ribozymes who can be on the 3' side, 3' untranslated region, in which case they might cut off a poly A tail at the end of the mRNA. And in eukaryotes, an mRNA that doesn't have a poly A tail will get degraded very quickly. So again, that's a mechanism for regulating the amount of protein that's being expressed from a mRNA. Also in eukaryotes, you can actually have changes in splicing. So you can have regulation of the splicing. And then the bottom here, figure 1D is sort of a special case of this sort of anti-termination and terminator mechanism, where you have transcriptional termination due to a riboswitch, but then, and I believe this is actually, let's see. Oh yeah, so if you have termination of the transcription, you'll be making these short pieces of the mRNA. That's just the part that contains the riboswitch. And in this particular case, that small piece of mRNA then actually also starts having a regulatory function in trans. So that small piece of RNA can sort of antisense bind to other pieces of mRNA and regulate their expression. So that's another cool trick. So some of the other things that aren't in this table here, I already mentioned that there's these temperature-sensitive mRNAs. You can think of those as riboswitches as well, where the regulator now is not a small molecule, but it's actually sort of sensing the temperature, and then the temperature has a change on the conformation of the mRNA that either induces or represses translation from the mRNA. And you can also have, obviously, you can have antisense-mediated regulation, where the regulator, again, it's not just a small molecule, but it could be another piece of RNA, right? So if you look at all these very complex folded RNAs, you can actually design a piece of RNA that's complementary to one of these stem loops, and if you add that piece, it'll disrupt the stem loop and change the conformation of this riboswitch, which could have the same effect as a binding of a small molecule to an aptamer, essentially. But that makes these things a lot more engineerable as well. because now you can target regulators using a piece of RNA or a piece of DNA, which is complementary to a specific set of bases in the regulator that you're targeting. So the equivalent of that, if you were to try and do things with proteins with transcription factors, is that you would have to have a transcription factor that you can target to a specific part of the DNA and affect like interaction with another transcription factor or something like that. Here you can actually target a signal to a genetic circuit by just having a little piece of complementary DNA or RNA that you can target to a specific regulator. I don't think I'm going to get too far into some of these other issues that are mentioned here in this paper. I wanted to mention one of the techniques that has been used to synthetically generate some of these riboswitches. So I mentioned in the beginning that people have known for a long time you can use mechanisms like CELEX to actually generate aptamers, to generate pieces of RNA that bind a specific small molecule of interest. And I think one of the other papers that I have in here has a nice figure on that. Right. So here's a paper that shows CELEX in action, where you essentially start with a large pool of double-stranded DNA, which is randomized, essentially, as a randomized region. You do in vitro transcription of that, so you generate a large pool of RNAs with a randomized region. And then you need to have a ligand that you're targeting that can be immobilized on a column. And you just pass all of these RNAs over that column, and the RNAs that stick to the ligand will have some affinity for it. And then you can elute the bound RNA, which will be a very small fraction of your initial pool. And you can do reverse transcription to generate, again, a DNA pool from that. And then maybe using error-prone PCR or something like that, maybe you can even add some more diversity if you want. But you can do multiple rounds of this. And essentially, you're starting out with a pool of random RNA sequences that might have, so your 10 to the 14 variants. And eventually, you wind up selecting out those RNAs that actually have a very high affinity for your small molecule of interest. And I've talked to some people that do this and that claim that they can pretty much generate the aptamers to whatever small molecule you give them within like a month or two. Just by running through this a couple of times, typically like 10 to 15 rounds of this selection to actually come up with an aptamer which has very tight binding to any small molecule of interest. So that gives you a way to make sensors for really any molecule, any metabolite or other small molecule that you want to measure inside the cell. So that's a very important tool for engineering more of these riboswitches. It turns out.
What paper was that?
This is from the TOPS ACS ChemBio. So that was it's the other review paper. Let me see what the title is.
Hey Patrick, did you send that?
Yeah, it's one of the ones that I sent out. So it's Emerging Applications of Riboswitches in Chemical Biology. TOPS and Gallivan, ACS Chemical Biology 2010.
Got it. Thank you.
So that's a very common technique that's been around for a long time. It turns out that actually using this to make riboswitches is a bit more tricky than just using this to generate aptamers. Because for a riboswitch, you also have to have a significant conformational change. A lot of these aptamers just have a specific shape that has an affinity for a small molecule, but it doesn't really change its shape upon binding the small molecule. And in that case, it may not be a very efficient riboswitch, because for a riboswitch, you typically have to do something like disturbing a stem loop that then reveals ribozyme binding site and stuff like that.
Quick question for aptamer use. So I know that they have aptamers where you have like a fluorophore and a quencher bound to either end, and I guess there are aptamers where it's just binding. So for this selection process, is it more typical to select for something that's just binding? Because I would think with a fluorophore and a quencher, you're going to undergo a big conformational change, right? I mean, for a binding.
Yes. Yeah. So here, everything is entirely passive. You're screening out from a large pool of variants. You're screening out the ones that actually stick to a column that has your ligand of interest, right? So yeah, it doesn't necessarily have any conformational change as long as it have enough affinity for the ligand. So one of the recommendations that they're making in one of these review papers is that you can do a lot of in vitro selection using Celex, but then you still need to do in vivo verification that this aptamer is suitable for making a riboswitch out of it as well. So there's various ways of doing screening for suitable riboswitches that are described in some of these other papers as well. All right. I think I'm going to leave it at that. Well, maybe one more. Let me show one more figure from this paper by Wynn and Smolke. Again, that's one of the other references that I have in the email that I sent out, showing how they're actually engineering some of these riboswitches. And this is one of these riboswitches that's based on a ribozyme, a hammerhead ribozyme that will cleave itself. And they're actually engineering this into the 3' untranslated region behind a gene, but in front of the poly A tail of the gene. So this is in eukaryotes, right? So when this ribozyme cleaves off the end of the mRNA, it destabilizes the mRNA, and the mRNA gets rapidly degraded. If that ribozyme is not activated, then the gene is expressed normally. And you're seeing how they're starting out with this in figure A here with this what they call the actuator. That's actually the hammerhead ribozyme. And you can see that it kind of looks like a hammerhead shark. That's why it's called the hammerhead ribozyme, as far as I know. And they figured out where to attach some of these aptamers to this ribozyme to actually affect the function of the ribozyme. So you see here, this is how you can add a single sensor, a single aptamer that senses the presence of a specific small molecule. And then they have various ways of extending that. You can have 2 of these hammerhead ribozymes in the 3' untranslated region. You can actually modify 2 different loops of that ribozyme with 2 different sensors, or you can actually sort of chain these sensors together. And again, this will form various different Boolean gates. In some of these cases, you need to have, you may need to have both of the small molecules present to inhibit the ribozyme or vice versa. So there's lots of engineering that can be done here, and it's all based on engineering RNA structures. And there's actually some pretty decent software available to predict RNA structures at sort of secondary and tertiary structure level. This is something that, in theory, should actually be much easier than engineering proteins to fold in a specific fashion, because RNAs are they have much less diversity in the units that they're using, obviously, right? So you only have to deal with 4 bases. There's only so many ways that these things can fold. So in theory, it should be much easier to predict how some of these RNA devices will fold and how a protein will fold. So that opens up a lot of possibility for engineering these things computationally and then testing them out in the lab. Also, if you look at how large some of these things get, this is still much smaller than actually engineering a protein. So you're talking about maybe a couple of 100 base pairs, whereas you're probably talking about a couple of 1,000 base pairs if you want to try and do this with protein-based genetic logic. So. All right, and I think I'll hold it to that on that note. Any more questions? Did I manage to put everybody to sleep?
No, Patrick, thank you very much. That it really helped my understanding of the paper.
Sure.
I think
Yeah, this is a really exciting field, and I think all of that studies assays, it's something that really opens itself up really well to sort of DIY bio. With more accessible assays, engineering some of these RNA structures could be a lot easier than trying to do the same thing with protein-based genetic circuits as well.
Mm-hmm.
I think you increased the standard for the journal cloud today.
Yeah, but I know you want to go even deeper, Dennis.
So, and Oliver is doing the next presentation, so he should be prepared to keep up to the standard.
Well, I'll try. Is that the epigenetics paper with the Lamarckian transmittance of information?
Yes.
Yeah.
Is that what we're doing? The mice trauma, that one?
It's interesting.
The inherited anxieties, that one?
Yeah, I think. Yeah, it's not. There's definitely not as much molecular data in that paper as this one. It's yeah, but it's a totally interesting topic, and I think you can speculate like crazy over the implications.
Oh, I just saw an article in Nature or News about epigenetic of also some psychology things. I will find. Yeah, I will forward it to you.
All right, so I'll do that if nobody has any other suggestions, and I'll also give a background on epigenetics and maybe do some of the molecular stuff in the background, which is the types of epigenetic modifications and that sort of thing.
That would be great.
And if somebody at some point wants to do a follow-up journal club on riboswitches, there's lots of really cool recent developments.
Yeah.
That would make great papers.
Yeah, I would love to hear. I wouldn't mind having another riboswitch paper, a more recent one, because it seems like a field that's been accelerating. Like, unlike other fields where 5-10 years isn't that far back. It seems like in riboswitches, all sorts of stuff is happening really quickly.
Yeah.
So.
And really, it's sort of, you can do everything just in RNA essentially, right? And it's easier to engineer than proteins. So.
I think that paper about hammerhead RNA would be cool.
That's a really cool one. The other one by Lux. This is actually a paper from the Arkan lab here. Versatile RNA sensing transcriptional regulators for engineering genetic networks. So these guys were using antisense RNA genes that they can then chain together. So this is where they're using a riboswitch to control the expression of an antisense RNA that then can go to control other riboswitches.
I think that'd be real interesting as well. All right, I'm going to stop the recording, guys. Thanks.
All right. Thank you very much for sticking out to the end.
Thanks again, Patrick.